Hello students, welcome to EPG Pathsala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on stoichiometry of citric acid cycle under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. To understand the function of citric acid cycle in production of NADPH and FADH for synthesis of energy. To examine the yield of energy by completely oxidation of pyruvate, the conversion of pyruvate to acetyl group, then the entry of those groups into citric acid cycle, which is also called tricarboxylic acid cycle or Krebs cycle after its discoverer Hans Krebs. The TCS cycle is a clever and elegant way nature has devised to cleave a resistant 2-carbon acetyl bond, usually a very difficult task since it would correspond to an alpha oxidation, a process not very efficient and almost impossible in a 2-carbon species. Earlier we have learned about metabolic oxidation converting the glucose to acetyl coenzyme A. Acetyl coenzyme A is probably the most common metabolite intermediate found in cell. Acetyl coenzyme A is an activated 2-carbon compound found in many central metabolic pathways, including the citric acid cycle, the glyoxylate cycle, fatty acid synthesis, fatty acid oxidation, isoprene metabolism, amino sugar metabolism, ketone body metabolism, and cholesterol biosynthesis. The term activated used to describe the compound comes partly from the nature of the high energy thioester bond in the molecule with delta Go of 31.5 kilojoule per mole energy. Acetyl coenzyme A is one of the most ubiquitous metabolite in biological system. Acetyl coenzyme A is also useful as a building block of fatty acids, amino acids, steroids, prostaglandins, nucleotide and other compound. When both pyruvate molecules are oxidized to 6-carbon dioxide via the pyruvate dehydrogenase enzyme complex and the citric acid cycle, and the electron are transferred to oxygen via oxidative phosphorylation, as many as 32 ATP are obtained per glucose molecule. In round number, this represents the conservation of 32 multiplied by 30.5 kilojoule per mole equals 976 kilojoule per mole, or you can say 34% of the theoretical maximum energy of about 2840 kilojoule per mole energy available from the complex oxidation of molecule glucose. We will discuss about stoichiometry of reaction involved in the TCA cycle. Albert Leininger has rightly stated that living cells are self-regulating chemical engines timed to operate on principle of maximum economy. So if you see the concept map of stoichiometry of tricarboxylic acid cycle, the 4 carbon molecules oxaloacetate that began to cycle is regenerated after 8th step of citric acid cycle. The 8th step of citric acid cycle are a series of redox, dehydration, hydration and decarboxylation reactions. Each turn of cycle form 1 GTP or ATP as well as the 3 NADH molecule and one FADH2 molecule, which will be used in further steps of cellular respiration to produce ATP for the cell. The reaction of the citric acid cycle can be divided into two distinct phases. In the first part, 2 carbon of citrate are oxidized to carbon dioxide, yielding an activated 4 carbon compound, that is succinyl coenzyme A. In the second part of the citric acid cycle, succinyl coenzyme A is converted back to oxaloacetate, thus making it possible to begin the process again. In each turn of the citric acid cycle, 2 carbons are added from acetyl coenzyme A and 2 carbons are oxidized as carbon dioxide. Thus, there is no net incorporation of carbon from acetyl coenzyme in each turn of the cycle. 2 carbon atoms enters the cycle in condensation of an acetyl unit, that is from acetyl coenzyme with oxaloacetate. 2 carbon atoms leave the cycle in form of carbon dioxide in the successive decarboxylation catalyzed by isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Interestingly, the results of isotope labeling studies reveal that the 2 carbon atoms that enter each cycle are not the one that leave 4 pair of hydrogen atoms leave the cycle in 4 oxidation reactions. 2 molecules of NADH are reduced in the oxidation of decarboxylation of isocitrate and alpha-ketoglutarate. Stoichiometry of citric acid cycle. 2 carbon atoms enters the cycle in the condensation of an acetyl unit from acetyl coenzyme A with oxaloacetate. 2 carbon atoms leave the cycle in the form of carbon dioxide in the successive decarboxylation catalyzed by isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. 4 pair of hydrogen atoms leave the cycle in 4 oxidation reaction. 2 molecules of NADH are reduced in the oxidative decarboxylation of isocitrate and alpha-ketoglutarate. 1 molecule of FAD is reduced in oxidation of succinate and 1 molecule of NADH is reduced in the oxidation of malate. 1 compound with high phosphoryl transfer potential, usually GTP, is generated from the cleavage of the thioester linkage in succinyl coenzyme A. 2 molecules of water are consumed, one in the synthesis of citrate by the hydrolysis of citral coenzymes and other is the hydration of fumarate. If citrate is added, the rate of respiration is often increased. The extra oxygen uptake is by for greater than can be accomplished by the complete oxidation of citrate. Since citric acid reacts catalytically in the tissue, it is probable that it is removed by a primary reaction but regenerated by a subsequent reactions. These were the words of Hans Crabbe and Johnson, an article in Enzymology published in 1937. Discovered by Hans Crabbe in 1937, he received the Nobel Prize in Physiology or Medicine in 1953 for his discovery, forced to leave Germany prior to World War II because he was Jewish. The citric acid cycle may be seen as a method nature has invented to couple oxidation of acetate with water splitting and use the extra reducing equivalents as energy supply. In one of the classic paper, Rocker has interpreted the catalytic cycle as an acrobatic design nature invented to catalyze the splitting of hydrogen and subsequent use of its reducing equivalents to yield energy that is basically similar to the process in chloroplast where water is split at expense of light energy. The NADH and FADH2 molecule can be converted to ATP in oxidative phosphorylation. Depending on how they enter the electron transport cycle, electrons from NADH yield 3 adenosine triphosphate per NADH and 2 adenosine triphosphate per FADH2. Converted in this way, one molecule of glucose yield total 38 ATPs. Regulation of TCS cycle is very important. The TCS cycle must be carefully regulated by the cell. The unchecked citric acid cycle could lead production of large amount of metabolic energy and wastage in form of overproduction of produced coenzyme and ATP. Three factors govern the rate of flux through the cycle: substrate availability, inhibition by accumulating product, and allosteric feedback inhibition of the enzymes that catalyze early step in the cycle. The overall rate of citric acid cycle is controlled by the rate of conversion of pyruvate to acetyl coenzyme A and by the flux through citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. These flux are largely determined by the concentration of substrate and product. The end product ATP and NADH are inhibitory and the substrate NAD plus and ADP are stimulatory. Additionally, mitochondria derived reducing hydrogens from organic substrates. Complete oxidation of pyruvic acid yields 10 reducing hydrogen ions for NADHH plus and 1 FADH2 able to reduce and formation of 5 hydrogen molecules. In principle, the equation for pyruvic acid oxidation looks non-stoichiometric with 6 hydrogen missing. Pyruvic acid has only 4 hydrogen. The problem is to find the entries for the missing hydrogens and also 3 hydrogen in form of X. Therefore, 3 water entering the cycle and satisfying the stoichiometry. The effective number of hydrogen supplied by pyruvate is 4 as if it were pyruvic acid. Referring only to the cycle, the correct stoichiometry of acetyl coenzyme A oxidation will be as below. Since the process yield 8 reducing equivalent, Leininger described the net reaction of stoichiometry of citric acid cycle with an entry of 7 hydrogen. 4 of the extra stoichiometric hydrogen in pyruvate oxidation comes from hydrogen molecule entering at the level catalyzed by citrate synthetase and fumarate. The third missing water molecule has not previously been discussed in the clear term. Actually, this hydrogen enters indirectly via the cleavage of succinyl coenzyme thioester corresponding to a hydrolysis reaction without the intervention of molecular water. The cleavage of the thioester proceeds via phospholysis of a hydrogen equivalent from orthophosphate. This event provides the required free energy drop to phosphorylate ATP. Therefore, this extra hydrogen entering the Krebs cycle is carried out by phosphate. Ultimately, one may consider that this hydrogen is loaded in the phosphate carrier by the time of ATP hydrolysis catalyzed by ATPase during process of energy transduction in biosynthesis, mechanical work, electricity generation, and formation of ionic and metabolic gradients. Stoichiometry for pyruvate conversion to acetyl-CoA. Starting from acetyl coenzyme to the stoichiometry of citric acid cycle is shown as follows. Starting from glucose via glycolysis, the stoichiometry is as follows through the citric acid cycle. The net reaction of citric acid cycle, the NADH and FADH2 molecule can be converted to ATP in oxidative phosphorylation. Depending on route they enter into the electron transport cycle, electrons from NADH yield 3 ATP per NADH and 2 ATP per FADH2. Converted in this way, one molecule of glucose yields 38 ATPs. In round number, this represents the conversion of 38 multiplied by 30.5 kJ per mole, total 1160 kJ per mole, or 40% of theoretical maximum of 2840 kJ per mole available from complete oxidation of a glucose molecule. This table explains the stoichiometry of coenzyme reduction and ATP formation in the aerobic oxidation of a molecule of pyruvate by citric acid cycle. NADH molecules are formed during formation of 2-acetyl coenzyme A, alpha-ketoglutarate, 2-succinyl coenzyme A, 2-oxaloacetate from 2-pyruvate, 2-isocitrate and 2-alpha-ketoglutarate and 2-malate respectively. Formation of NADH molecule ultimately contributes to formation of 24 ATP molecules. 2 FADH2 molecules are formed during conversion of 2-fumarate to 2-succinate, leading to generation of 4 ATP molecules. Similarly, 2 ATP or GTP molecules are formed from 2-succinate from succinyl coenzyme A. Total ATP formed during the whole process is 38 molecules. Overall equations and stoichiometry. The 3 NADH plus 3 proton formed a turn of the cycle. Approximately 3 ATP formed NADH plus H by electron transport and oxidative phosphorylation. 1 FADH2 formed, 2 ATP formed per FADH2 by electron transport and oxidative phosphorylation. 1 GTP forms. Total equivalent of 12 ATP are formed and 2 turn require 2 acetyl coenzymes results in an equivalent of 24 ATP per 2 acetyl coenzymes. The table explains stoichiometry of TCA cycle by chemical equation, different enzymes involved and free energy release or gain in kilojoule per mole. Table shows the net free changes in the energy during citric acid cycle. This table explains different types of enzymes, prosthetic groups required during TCA cycle, reactions and of energy released or gain in term in kilojoule per mole. Reaction types are condensation reactions, dehydration reactions, hydration reactions, decarboxylation reactions, oxidation reactions, and substrate level phosphorylation reactions. The table shows the steps that I just discussed have been shown in this following chart here. The citric acid cycle must be carefully regulated by the cell. The unchecked citric acid cycle could lead production of large amount of metabolic energy and wastage in form of the overproduction of reduced coenzymes and ATP. At the same time, if the citric acid cycle ran too slowly, ATP could not be generated fast enough to sustain the cell. By looking at the change in free energy of the reaction of the citric acid cycle, it is clear that there are three irreversible steps. These three reactions of the cycle are citrate synthetase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase operate with large negative charges free energy changes under the concentration of product and reactant in the matrix of the mitochondria. The TCA, in contrast, is regulated by three simple mechanisms: substrate availability, product inhibition, and competitive feedback inhibition. The allosteric effector that control the flux of metabolic through the TCA cycle are acetyl coenzyme A, succinyl coenzyme A, ATP, ADP, AMP, NADH, and NADH plus NAD plus. Figure shows the key points where reactions are regulated. Regulatory steps are pyruvate dehydrogenase complex, ATP, acetyl coenzyme A, and NADH inhibition. Second is covalent modification by phosphorylation. And third require TPP, FD, lipoic acid cofactors. Citrate synthetase inhibited by ATP and citrate, also NADH and succinyl coenzyme A. Isocitrate dehydrogenase inhibited by ATP, activated by ADP and elevated NAD-NADH ratio. Alpha-ketoglutarate dehydrogenase. This enzyme is inhibited by ATP and high succinyl coenzyme A. It is activated by ADP and elevated NAD-NADH ratio and coenzyme A succinyl coenzyme A ratio. Require cofactor TPP, FD, lipoic acid. Cells with low energy requirements have high ratio of ATP and ADP and NADH and NAD concentrations ratio. Cells with very active and significant energy use have low ratio of ATP and ADP and NADH and NAD concentration. If we see the concluding remarks, the TCA cycle, also known as citric acid cycle, is a universal central metabolic pathway in which compounds derived from the breakdown of carbohydrates, fats, and proteins are oxidized to carbon dioxide, with most of the energy of oxidation temporarily held in the electron carrier FADH2 and NADH. Acetyl coenzyme A enters the citric acid cycle in the mitochondria of eukaryotes, the cytoplasm of prokaryotes, as citrate synthase catalyze its condensation with oxaloacetate to form citrate. In 7 sequential reactions, including 2 decarboxylation reactions, the citric acid cycle converts citrate to oxaloacetate and release 2 carbon dioxide. The pathway is cyclic in that the intermediates of the cycle are not used up. For each oxaloacetate consumed in the path, one is produced. So students, let's summarize what we have learned in this module. The citric acid cycle is a central metabolic pathway which generates NADH and FADH2 for use in electron transport. It also produces GTP via substrate level phosphorylation. Many metabolic processes use intermediate of the citric acid cycle in their pathways. The cyclic process is generally considered to begin with additional of acetyl coenzyme to oxaloacetate to form citrate. So student, this is all about stoichiometry of TCA cycle. Thank you.